last time we sort of finished up the story of DNA methylation. And what I'd like to do today is tell you a little bit about what we know about DNA methylation and cancer. So this is not really basic research, but you'll see that there's basic aspects to this that are probably very important for our understanding of this process. Okay, so a long time ago, maybe 20 years ago, it was discovered that there are regions of DNA that undergo de novo methylation in a number of different tumors, a number of different types of cancer. And of course, as you would imagine, the regions that undergo de novo methylation have to be CpG islands, because it's the CpG islands that are unmethylated to begin with. So if something undergoes de novo methylation, it has to be a CpG island. And so again, just to put it into perspective, these things are left open, or left unmethylated from early time in development. They remain unmethylated throughout development. It doesn't mean that they're all turned on, all these genes that are unmethylated. Some of them are repressed by other mechanisms. But they remain unmethylated throughout development. And in tumors, some of these CpG islands now get methylated. Okay? And that's abnormal because they're not methylated anywhere in the body. So that makes it an abnormal methylation. It seems to be associated with cancer. So at the beginning, before there were global methodologies for looking at DNA methylation, people were interested in tumor suppressor genes. And the idea was that if something gets, well, the idea is that in tumors you find that tumor suppressor genes are inactivated, usually by mutation. So if you take a look in every colon tumor in man, for instance, there's a gene called APC. Every single, or almost every single colon tumor that you see has APC inactivated or mutated in the tumor. Okay, so this is a typical tumor suppressor genes, and there are many tumor suppressor genes of this nature. So when they started looking at these genes, it was discovered that sometimes you find that only one allele has undergone mutation or some sort of change. So the question is, what about the other allele? And so they started looking at methylation, and sure enough, you find that many times these tumor suppressor genes are methylated in the second allele. So this sort of brought up the idea that methylation plays a role in cancer by inactivating tumor suppressor genes. How does it work? The idea, the dogma was, or the concept was, that it works similar to a mutation. That mutations work in the following manner. Let's say you're exposed to something that causes mutation, or you have something in your genome that makes repair not so good. And you get a mutation. Now the mutations are basically random. They can happen anywhere in the genome. Maybe not completely random, but they can happen in a lot of places in the genome. Right? But because there's selection, because cancer cells grow, there's selection, you end up seeing the mutations basically in the tumor suppressor gene. Okay? And they said, well, maybe this is the way methylation works also. In other words, the same concept. You have something happens in cancer, in a tumor cell. There's a leakage of de novo methylation. Remember, usually you don't get de novo methylation in somatic cells. But they're not undergoing active de novo methylation. Right? So maybe there's some sort of leakage. There's excess de novo methylation. The de novo methylation methylates randomly, but there's selection. If it methylates a tumor suppressor gene, that will be selected for. So this was the concept of how de novo methylation plays a role in cancer. Okay? So I'm going to tell you about something that was basically started, the thinking about this thing started when Itai was in the lab. And we came up with so this just shows you what I just told you right now. That people thought that I forgot to bring my pointer. People thought that if this is a tumor suppressor gene, so one allele gets mutated, the other allele maybe gets methylated. Okay? So Itai and I started thinking about this question, and we asked ourselves the question of whether this process, rather than being a selective process, might be an instructive process. In other words, maybe de novo methylation is not the way we've described it, but maybe there's a process for de novo methylation that's an inherent part of the cancer genome. And it has nothing to do with selection. Certain genes get de novo methylated. Now, I want to tell you what led to this concept, because this is a general idea in biology. Okay? What led to this basically started off from what I think was a fantastic experiment which was carried out by Charles Emerson. And he did the following experiment. He grew 10 T1/2 cells. 10 T1/2 cells are sort of fibroblast-like cells. He grew them in culture. And he found that if you treat these cells with 5-azacitidine, which is a demethylating agent, if you treat these cells with 5-azacitidine, you see that cells undergo differentiation. And they undergo differentiation in 3 different directions. So you take cells treated with, you take 10 T1/2 cells, you add 5-azacitidine, a lot of cells die, but you end up with seeing colonies that have undergone differentiation. And you always see differentiation in one of 3 directions. Either you see differentiation to muscle cells or to chondroblasts, schus, how do you say that? Cartilage, or you see to adipocytes, to fat cells. Okay? And you always get these 3. Most of them are muscle phenotype, but you also get the other phenotypes as well. And Emerson asked himself the question, how does this happen? Because normally you would think, okay, you take a cell, or you take cells, you treat them with 5-azacitidine. 5-azacitidine causes demethylation. So you get demethylation, you figure it's random. And every once in a while you get just the right combination so that it'll make a muscle cell. Or you get just the right combination of turning on genes and you get an adipoblast. And Emerson said, this doesn't make sense. Because what did he do? He took these cells, each one of them. He took the muscle cells, and he analyzed how many new proteins appear in the myoblast cells that he got. And he found that it was something like 50 to 100 new proteins. He took the adipoblasts and did the same thing. Also, 50 to 100 new proteins appear in these cells. So, and it's always the same. Every clone has the same proteins being induced. It's a real adipoblast. So he asked himself, it can't be random. What's happening here can't be random. When you get a muscle cell, it can't be just random demethylation of genes. There has to be something directing it. Right? And he was basically the one who came up with the idea of what we call today a master gene. He was the one who initiated the idea that led to the discovery by Hal Weintraub of MyoD. MyoD is a muscle master gene. And in fact, what was happening in these cells was that 5-azacitidine was turning on MyoD, right, which is this one gene you have to turn on. And once that got turned on, it influenced all the other genes that you need for muscle, to make a muscle phenotype. Right? And that's the way it worked. It worked on the basis of programming. There's a master gene which programs the gene expression profile of the cell. Okay? And so this was a very, very important concept. By the way, this was all, of course, an artifact. Because MyoD in vivo is a CpG island G. It's never methylated. But in cells that grow in culture, like 10T1/2, MyoD got abnormally methylated, and that's why this 5-iso-C experiment works. And the same thing is true, of course, for the chondroblasts and the adipoblasts. Eventually, somebody did find a gene, AdipoD. It works the same way. There's a master gene. Okay? And this is a general biological phenomenon. When you see cells that now adopt a new phenotype, and this phenotype is clearly not the result of one single gene, or involves numerous genes, numerous changes, chances are that this involves some sort of master regulator. And all this went into the thinking that Itai and I had, and we came up with the idea that perhaps in these tumor cells that's what's happening, that there's some sort of master regulator that causes methylation at certain places. And indeed, that's what turned out to be the case. So if you now, Ilana developed this assay, as I mentioned before, for looking at methylation throughout the genome. And if you now take tumor cells, these are colon tumors, fresh tumors. As you realize, you can't take cells in culture. Cells in culture are artifacts in terms of methylation. These are real tumors. Okay? And you can see here there are many, many, many CpG islands that are unmethylated in every tissue, clearly unmethylated all over the place. These are clear CpG islands that are slated to be unmethylated from early time in development, yet in tumors they're methylated. And this is what you see here. If you could scroll down on the slide here, you would see that there are about 2,000 of these. So these colon tumors have almost 2,000 CpG islands. They're not all at promoters. CpG islands that get de novo methylated in the tumor. Okay, so this is a lot. This is hard to explain, really hard to explain on the basis of selection. Okay? Incidentally, it could very well be that mutations, this model started with the mutation idea, it could very well be that mutations are also not like that, that there's some sort of programming that goes into mutations. And now that people are sequencing all sorts of tumor types, there's a picture like that emerging, that in tumors, every tumor type has certain genes that get mutated. It's not, may not be completely random. Okay. Now, it's not a surprise, but these genes turn out to be marked. A large percentage of these CpG islands are marked with polycomb. They're polycomb target genes. They have EZH2 on them. They're methylated in lysine 27. They're heterochromatinized by the polycomb group protein, which binds to methylated lysine 27. Okay. And this is actually this was an early estimate. Later estimates indicate that as many as 70 or 80% of these CpG islands are actually targets for polycomb. So polycomb is probably somehow involved. We know also that EZH2, which is the center of the polycomb complex, has the ability to recruit DNA de novo methylases. Okay? So it makes sense that in the tumors, genes that are labeled with polycomb are prime targets for de novo methylation. What triggers the de novo methylation? We don't know. In a minute, I'll tell you a little bit about that. Right? But it seems like what gets methylated is indeed programmed. And it's a simple program in the sense that it's polycomb targets. So the programming involves we don't know how polycomb is recruited to these CpG islands yet. Right? And the computational people have not yet come up with a sequence. I'm looking at specific people here, well, like Yoav. You could just figure out how what the sequences that are involved. Might not be just sequences as well, but somehow these get targeted to polycomb, and that's the source of this de novo methylation. Incidentally, if you now look at the genes, when you now look at promoters that are getting methylated, okay? So the original concept of selection was that the idea was that in the normal tissue, these genes are active. And they get de novo methylated, maybe by accident. Right? But they get de novo methylated, and that inactivates them, and then you get selection. That was the original idea. Okay? Now that we know all these CpG islands get methylated, you can go back and look at the transcription picture. And you see that the ones that are getting methylated, over 90% of them are not active in the colon to begin with. They're silent. That makes sense, because most of them are polycomb targets. Polycomb is a repressor. These genes are repressed. Most of them are repressed in the colon to begin with, and then they get methylated. So there's clearly no selection involved here. The idea was correct. Okay, so now this led to the following picture, which is a picture, it's a speculation of how de novo methylation may play a role in tumorigenesis, and I really put the emphasis, put on underline and quotes. This is a speculation. Okay? The idea is as follows, I think. It's based on the idea that they're polycomb targets. So this is the colon. This is the epithelium of the colon. The colon is made up of crypts. In the crypts, there are stem cells at the base of the crypts. These stem cells every once in a while divide. They generate a proliferative progenitor and another stem cell. Every time this cell divides, it makes another stem cell, and it makes this proliferative cell. This replicates and divides rapidly. It moves up the crypt. When it gets to a certain point, morphologically, it undergoes differentiation to epithelium. Okay? That's how the colon is generated. Okay? And this, of course, occurs all the time. It's occurring now within us. This is a turnover. About every 4 days, we replace the colon, the epithelium of the colon. This is an active process always going on. So the idea is as follows, that that methylation contributes to the tumor by affecting differentiation. And I'll explain to you the idea. The idea is that this is one of these genes that is getting de novo methylated. Okay? In the colon, in the normal tissue, in the stem cell, and the progenitor cell, and in the proliferative cell, right? Of course, this gene is unmethylated. It has the polycomb complex, EZH2. Because of that, lysine 27 is methylated. And the gene, of course, is repressed. Not repressed by methylation, repressed by polycomb. This in itself, of course, this attracts a chromodomain protein called a polycomb group protein. And that heterochromatinizes it. That's one of the things that makes this repressed. Okay? At the time of differentiation, some of these genes, which are required for differentiation, remember, they're polycomb targets. Polycomb targets are the developmental and differentiation genes of the genome. All of the important genes that are involved in differentiation or development are polycomb targets. They're usually repressed throughout development, from the beginning of development. And then, when they get activated, like MyoD, MyoD is polycomb everywhere in the organism, but in the muscle cells, if you look at the MyoD, polycomb has been taken off. That's the way the system works. Repressed everywhere, it gets activated in muscle. Okay. Some of these genes are required or used for differentiating to the epithelial cell type. So those genes now lose their polycomb. They lose their polycomb, they get activated. The histones get acetylated, lysine 4 gets methylated. This gene is now active. So this is what normally happens to these genes that are involved in the differentiation of colonic tissue. Okay? Our hypothesis is that already in the stem cells, in order to form the tumor, already in the stem cells, you have methylation. Okay? You have DNA, it's gotten methylated. Okay? Now that's not going to affect very much in the proliferative cell. Because in the proliferative cell and in the stem cell, these genes are repressed to begin with. It doesn't make much difference if they're methylated. But now when this gene tries to get activated, okay, it may be able to take off the EZH2. Chances are it does. But because the gene, the promoter, is methylated, it can't turn the gene on. And if it can't turn the gene on, it can't differentiate. And if it doesn't differentiate it, it remains a proliferative cell. And this idea that it not differentiating, we know that lots of times in tumors, differentiation is an important factor. And here probably is one of the key factors. You end up with a cell that now has a proliferative phenotype. It may not be a tumor yet, but it certainly contributes to the tumor phenotype. You probably need something else. Okay, so this is the idea. Consistent with the idea are a couple of things. First of all, just to point out to you some of the polycomb target genes. You can really see that these are genes that are involved in differentiation. All these MyoD, Wnt, whatchamacallit, SOX genes. Right? This is important for the liver, for neurons, for morphology. All these important genes are all target genes of polycomb. And we know some of them, we know at least 10 of them. I didn't list them here, but there are at least 10 of them. This, for instance, Wnt10A, for instance, are involved in differentiation of colonic tissue as well. Okay, just to give you... So in support of this idea is this experiment by Rudolf Jaenisch, which was done more than 10 years ago, maybe 12 years ago. And what he did was he took mice that are a model for colon cancer. These are min-minus mice. They have one allele of the APC gene, which in the mouse is called min, is deleted. And they develop tumors. They develop somewhere between 75 to 100 tumors per mouse, which you can see at the age of 100, about 3 months, a little bit more than 3 months. So for instance, a normal mouse, if you look at the number of adenomas, intestinal adenomas that develop, the normal mouse has about 75. When you wait longer, it gets up to 100. Right? So what Rudolf Jaenisch did was he treated these mice from birth with low doses of 5-azacitidine, which inhibits DNA methylation. So he started every week, once a week, he gave a low dose of 5-azacitidine. And the result is that if you now look at these mice, okay, that got 5-azacitidine from birth, you get very, very, very few tumors. Okay? A really very effective way to prevent this tumor. Now, pay attention to the fact that, first of all, these tumors don't arise without the APC deletion. But there's a genetic component here. Without that, you don't get the tumors. Okay? But this is a mouse. This is like a woman who has the BRCA1 gene. Right? She has an 80% chance of getting tumors. These have a 100% chance of getting tumors. Right? But yet, by preventing this methylation or inhibiting methylation, it prevents the appearance of the tumor. Okay? So this says that you need basically two things to get these tumors. You need the methylation and you need the mutation. Okay, without the mutation, you don't get the tumor, and presumably, by this experiment, without the methylation, you don't get the tumor. Okay. This is just to give you if you look, these are what's called aberrant crypts. If you look in these animals, you see these aberrant crypts. These aberrant crypts are not tumors yet. Right? These aberrant crypts are caused by the loss of the second APC gene. Okay? So again, these animals are heterozygotes. They're missing one allele. But through their lifetime, they develop mutations in the second allele. And as a result, many of the crypts, many of these things, by the way, have to happen in stem cells. Right? I mean, if you get a mutation in the epithelial cell, it has no significance. That cell is going to die. Right? All of these things have to happen in the stem cell. So if you have a mutation in both alleles, you get this thing called an aberrant crypt. Right? This is basically the equivalent of so this is what the mutation does. Right? The mutation causes this. And if you take a look at animals that were treated with 5-azacitidine, you still see the same number of these aberrant crypts. It's still making these aberrant crypts. The mutation's going on as usual. And it's making these things. But these are not tumors. Okay? Without the methylation, this cannot develop into a full-blown tumor. Okay? So this gives you a picture for the relationship between these two aspects, the genetic aspect and the epigenetic aspect. And this may very well be true for many, many tumors. Okay, now I just want to give you an idea. This is the work of Dvora, just to bring you up to date. The question is, where does the methylation come from? Where does it start? Where does it come from? Right? And so what Dvora did was she took all the 2,000 CPG islands that undergo de novo methylation in tumors look like this on a Z-score map. Okay? So this is Z-score. Lower numbers means less methylated. Higher numbers means more methylated. Okay? These are all the CPG islands that are constitutively unmethylated. So they have an average of very low Z-score. They're unmethylated. These are all the CPG islands that are constitutively methylated. They have a high Z-score. So these are our controls, or standards, right? Unmethylated, methylated. And this are all the CPG islands that are methylated in the tumor. This is what the tumor looks. So it's not fully methylated, right? But they're highly methylated. Right? And then Dvora looked at normal colon, at the same genes. Okay? And this is what it looks like in the same exact genes. The ones that get methylated in cancer look like this. Okay? So you can see in the colon, this is a colon of about a 60-year-old patient. You can see that they're already methylated. They're not methylated a lot, but they're clearly already methylated. So it looks like a lot of the methylation is already present in the normal colon way before the tumor gets born. And this is the whole colon. This is a normal colon. Right? We're not looking at one specific place that's going to turn into a tumor. This is the whole colon. Right? Already methylated. How does this methylation come about? So if you look at a young person, it looks like this. You look at the 60-year-old, it looks like this. It looks like it increases with age. So it's something that occurs during age, during aging. Okay? This is just to show you what it looks like in ES cells. The same population in ES cells is completely unmethylated. Okay? So it starts off, these sequences that are prone to methylation in colon cancer start off unmethylated, an increase in methylation as a function of age. It's highly tissue specific. So this is what I just showed you, the genes that get methylated in colon cancer. Okay? If you now look, and this is what they look like in normal colon, this is what they look like in brain, and this is what they look like in muscle. Much less methylated. Okay? So it's tissue specific, this methylation. Here's another example. This is just shows you, this is taken from, these are CpG islands that undergo methylation in hepatomas, in liver tumor cells. In the liver, they're already methylated, in normal liver, and it's tissue specific, not in brain, or less in brain and less in muscle. Okay? So there's a process, a slow process. We don't know much about it. There's a slow process of de novo methylation. It's attacking polycomb genes in general, but there's also a tissue-specific component which we really don't understand yet. There's something about the factorology of the colon that brings about slow de novo methylation in the specific tissue colon. And again, I emphasize this process has to be happening in the stem cells. It must be happening in the stem cells, because those are the only cells that are basically long-living. Okay, I think we're going to stop here. And I just want to point out that this doesn't look like a lot of methylation. Okay? There's not as much as in the tumor. But if you take a look at these genes, if you do now bisulfite sequencing, and look at these, the molecules that you get, right? And you ask how are they methylated. So you take a normal colon and you bisulfite sequence these particular CpG islands, you see that there's a bimodal distribution. There's some of the molecules are not methylated at all, or very, very little. And the other ones, so when we say this is 20% methylated, it almost means that 20% of the molecules are fully methylated and 80% not methylated. I'm exaggerating, but that's the idea. Okay? So there seem to be pockets in the colon, in the normal colon, that are highly methylated, and those could very well be the source of the tumors. Okay? That's where we stand on the tumor. I really brought this story because I wanted to talk about the whole concept of programming. Okay, now give me a half a minute, and we're going to switch over to what did I write here? This shouldn't be methylation. This should be replication. We're going to switch. That's not right. Wow, it's not here. Okay, give me a second. I'll get it. One second. Okay, now we're going to switch topics, and I want to talk about something else that I think is also a so-called epigenetic, an epigenetic mechanism. Okay, and I want to put this into perspective. Okay, DNA methylation is an epigenetic mechanism, according to my view, because it is a mechanism for preserving chromatin structure through replication. Okay? So a gene that's methylated, or a region that's methylated, there's a mechanism for copying that methylation onto the DNA copy, and then that methylation can serve as a mechanism for rebuilding chromatin structure after replication. Okay? Another mechanism for rebuilding chromatin structure after replication is a simple transcription factor. As long as the transcription factor is expressed in the mother cell and expressed in the daughter cell, okay? So originally in the cell it's binding to some sequence and doing what it does, either repressing or activating. After replication, that factor is still present in the cell. It can again bind and do the same work. So that's also sort of a mechanism, and many times these factors affect chromatin structure. They cause acetylation or deacetylation or methylation of something. So that's also a mechanism. Methylation is a mechanism that's also stable. So even if the original factor goes away, it will remain. It's a little bit different. What I want to try and sell you here, and it's going to be a sell, not easy, is that replication timing is another epigenetic mechanism like methylation. It's long-lasting, and it helps rebuild chromatin structure after replication. Okay, so first, a couple of important facts. So if you take a look at the genome, it turns out that it doesn't undergo replication equally all over the chromosomes, all over the genome. Rather, different regions of the genome undergo replication at different times. Okay? And you can see that in this experiment where you take cells and you label them with BRDU for 2 hours, and then after the labeling period, you immediately look at cells in mitosis. So if I do that experiment, take cells that are not synchronized, I label for 2 hours, but then I only look at cells in mitosis. So I'm only looking at what you see here, the cells that were labeled here. Okay? So here it's been labeled for 3 hours, I'm sorry. And then we look at mitosis. You see the following picture. This is a typical chromosome. You see 1 or 2 regions labeled with BRDU. Okay? That means that this region underwent replication over here, towards the end of S. Okay? Because we're only labeling, we're only looking at labeling in the end of S. So when we see this, this has to have been replicated towards the end of S. Okay? If we now label for a longer period of time, we pick up new regions. So the original region, we pick up new regions, because these regions replicate at this time in S. It's still late S, but earlier than this. And you can keep doing this experiment and in that way map replication timing regions or zones. Okay? Now, pay attention to the fact that it's indeed regional. Okay? You might have expected that if you do an experiment like this, you just labeled the last hour of S, you might see label all over the chromosome, a little bit of label all over the chromosome. You don't see that. Replication timing, the time when something replicates, is controlled at a regional level. And that, in fact, defines chromosome bands. The usual method for looking at chromosome bands is what's called G bands, labeling with Giemsa. You do trypsin and Giemsa. Nobody really knows what this labels, right? But it picks up features of the chromosome, structural features of the chromosome. The Giemsa bands represent the open regions, basically the open regions of chromosome. Right? Late replication are the opposite of that. Okay? So what replicates late is a non-Giemsa band. What replicates early is a Giemsa band. Okay, so basically this is just a schematic, shows you how the whole genome gets copied. It gets copied in regional manner. So first, let's say this region over here gets replicated in early S, like that. Then this region gets replicated. That's second quarter of S. And then another region gets replicated in the third quarter of S. And finally, the entire chromosome gets replicated and picks up the late S. So this is what replication looks like in a schematic manner. Replication timing looks like. And as I mentioned, these regions correspond to G bands. So when you see a banding pattern, this is basically the same as the banding pattern of replication. And the bottom line is that chromosomes are divided up into units. And these units of about 1 to 2 megabases in length represent some sort of structural unit, as detected by Giemsa. They represent a replication timing unit. That small unit replicates at a specific time. And they represent some sort of activity type unit. The regions that are early replicating, for instance, are more open, they're DNA sensitive and active, whereas regions that are late replicating are less open and relatively inactive. So there's a correlation between replication timing and gene expression. This is a very old experiment. I used to say it's 25 years old, but I think by now it's 30 years old. This is an experiment that was done by Hübner, in which he looked at origins of replication. Okay? So he took DNA, he took cells, he labeled them for a short time, a couple of minutes, 1 or 2 minutes, with tritiated thymidine. Okay? Then he took the DNA from the cells and he laid it on a grid for looking in the electron microscope. And then he put it away in the dark room for 6 months to a year, exposed to film, and after the 6 months or a year, he takes a look at the exposure on the film, and you see the following thing. Okay, so let's take a look over here. This one's okay. So, first of all, you see all these black lines are regions that have undergone replication and incorporated thymidine. Okay. What is this? What are we seeing here? What we're seeing here is that at the center of each of these lines is an origin. Okay? Now, this region replicates at some specific time. Okay? And he caught these cells in the middle of replication. They already started replication. Right? And he caught it in the middle. So what happened? There's an origin right in the middle here. There's bidirectional replication. And you get replication like this and like this. About 100 kilobase downstream, there's another origin. And that does the same thing. Okay? And here there's another origin. Again, the same thing. Another origin over here. Okay? You notice that this whole region replicates at the same time. This gives you a picture of how these replication time zones are replicated. You have a replication time zone. It can have 10 or 20 of these origins, and they're all coordinated to replicate at the exact same time. And that's what we're seeing here. Right? This coordinated replication. This one, by the way, if I understand it correctly, here he caught this region of the genome was caught after it had already initiated replication. The origins had already started. And he added the thymidine about a minute or two after these origins had started. So what you see is this. Here, the origin is here. Okay? It had already proceeded this way and this way with no label. There was no label present yet. And then the label was added, and you get this replication. And here's, again, origin, replication to the two sides. Origin, origin. I don't know what happened over here. Here's another one. Okay? You can't, these are actually much longer regions, but you can't see them because, you know, the DNA breaks and you don't always see these things. But clearly, the point I want to make is that all these origins are in a replication time zone, and the firing of the origin, the starting of replication, is coordinated within a time zone. Okay, so now you have a picture of how the genome is organized in terms of replication timing. There are big regions, 1 to 2 megabase. Each one replicates at a certain time. We don't know what controls that yet, but we're going to talk about that. And it works by initiating the firing of a whole bunch of origins in a coordinative manner at the same time. Okay. Now, you can also measure replication timing. What's the next one? Yeah. You can also measure replication timing of a particular gene. Then I mentioned that there's a correlation between replication timing and gene expression. Early replicating genes tend to be active. Inactive genes tend to be late replicating. And it came out of studies like this. So, for instance, so what you do here is you take cells, you label them with BRDU for an hour. Okay? So now the label has gone in all sorts of places in the genome. You now take the cells and you divide them up into what stage of S they are when they got the label. Okay, so you do this. There are two approaches to doing this. There's a machine called an elutriator, which separates cells on the basis of size. Or you can do FACS. Again, separate cells on the basis of size or on the basis of the amount of DNA they have. And in this way, obtain cells from very early S, cells from the second quarter of S, cells from third quarter of S, or you can divide it up into 8 if you want, or 10 if you want. And you get different parts of S. Then you take each one of these cells, you isolate the DNA, and you selectively immunoprecipitate the BRDU DNA. Okay? So now I have in test tubes the BRDU DNA from early S. So this contains all of the genes that replicated in early S. Next fraction, I have the BRDU DNA from the second fraction of S. This fraction of DNA contains all of the genes that replicated in the second part of S. Okay? So now what you do is you take these fractions and you assay them for a specific gene. Okay? You can do this by blotting. You can do this by PCR. Okay? And you get results that look like this. So here's a gene, let's say ICAM5, and I don't remember what cells these are. But here this S phase has been broken up into 6 fractions. Okay? And you can see this gene replicates in early S. Okay? Because only the first 2 fractions have that gene in their BRDU. So they replicated in early S. Whereas this gene in the same cells replicates in late S. You only see it towards the end of S. Okay? Maybe this replicates sometime in the middle. So this is a way of looking at replication timing of an individual gene. And from these experiments came out the idea that there's a correlation between early replication and gene expression. I just want to show you, I might go a minute or two over, but I want to show you the assay that we developed in our lab. And the assay was based on this picture in Science. Okay? I love to tell this story, so and probably many of you have heard it, but I'm going to tell it again. So anyhow, David Ward published this paper. This was the first time that somebody, up until this, people had looked at the location of genes on chromosomes, but they had used radioactive materials. Okay? And the positioning is not very active when you use radioactivity. You don't have to wait 6 months, but still, it's not accurate. And he developed a system of using dye, fluorescent dyes, something we take for granted today. We do it all the time. But he developed this system of what we call FISH. Okay? And what you see here, I believe this is the globin region on chromosome 11 in humans. And you see two of the chromosome 11. And you see the reaction to the genes. And of course, this is at mitosis. So each of these chromosomes contains 2 copies, 2 chromatids, 2 copies of every gene. And you see that, right? And of course, the other one as well. Okay? So I was sitting with Sarah, we were looking at this, and we came up, we said, wow, if we could look at these dots in the nuclei, we might be able to have an assay for replication timing, because if an early S cell should have single dots and later S cells should have double dots, and maybe we can use this for replication timing, right? So we immediately went back to this picture and we looked, and here's a nucleus in the picture, but you don't see any dots. Okay? And so we thought maybe this method doesn't work on interface cells, right? Who knows? We had no idea. So fortunately, David Ward was a good friend of mine. I called him up on the telephone, and I said, David, we're looking at your picture. I said, why aren't the nuclei labeled? So he said to me, oh, they are labeled. I said, but I don't see it on the picture. He said, oh, this picture? One of the alleles is located over here under the S, and the other one got cut off on the top. So indeed, you can see it on interface nuclei. And this makes a wonderful assay, a very accurate assay. So it looks something like this. These are cells that are in S because they're labeled, background labeled with BRDU. So we know these are cells in S phase. And when you look in S phase for any probe, you always see 2 types of cells. You see cells that have 2 single dots, meaning that they haven't replicated yet. This gene hasn't replicated yet. And you see cells with double dots, meaning that the gene has replicated. Okay? And so it's very easy now. You can just count. If you take a whole bunch of nuclei, and you say 100 nuclei, and you now count how many singles and how many doubles, if the gene is early replicating, okay, most of the cells will have double dots. Okay? Because it replicated already, the earliest cell has a double dot. And of course, the latest cell will also have a double dot. Okay? Whereas if the cell is if the gene is late replicating, there'll be few double dots and a lot of single dots. And you can actually quantitate this, and it provides a very accurate assay for replication timing, as basically shown by this slide. Can everybody see it? Sort of? Okay, so this is a double labeling experiment, 2 different genes. Okay? And when you do this, you get 3. Okay? You get nuclei that look like this, in which one of the genes, the yellow gene, the white gene, has not replicated, and the red gene has not replicated. Okay? You get nuclei like this, where both the white gene and the red gene have already replicated. And then you get nuclei that look like this, where the white gene hasn't yet replicated, but the red gene has replicated. So this says that red replicates before white. Okay? And every cell, you never see the opposite. You can't see the opposite because it's programmed. The red gene always replicates before the white gene. Okay? So this sort of shows that the assay is accurate and picks up this programming. So in next session, we're going to try and understand all the aspects of replication timing. It's not simple. There are a lot of aspects to address here. We have to ask ourselves the question, what controls replication timing? Once we know what controls replication timing, we have to know how that impacts on the origins. Okay? We want to ask ourselves the questions, what the relationship between replication timing, how do you explain mechanistically the connection between replication timing and gene expression? Right? So we're going to, all these questions have to be answered, and we're going to try and do that. Okay? I don't promise answers to everything, but we're going to at least try. Okay, thanks a lot.